Ready or not, here we come.
See, this is when they're feeling generous. I just want to see which one he would pick.
It's a big one.
Good.
Overcompensation.
It is my pleasure to introduce Stephen Morgan, who has come here today with a little change in schedule, as all of you, from UC Davis. So I want to thank Stephen for being flexible. And I'm going to start out by telling with a quote, which is, "Blessed are the flexible, for they won't be bent out of shape." So, because Stephen sums that up in both his outlook, I think, and also his look into science. So he has come here from UC Davis. We are sorry about the change in time. We have a lot of people in the field and various field trips, so I'm glad that the marine ecology class could make it, because it's pertinent to both Stephen and his work is pertinent to your work. He has, I first met you when you were the visiting scientist at Moss Landing in
the glory days.
Glory days, 2000, I think? Maybe?
Yeah, maybe.
But Stephen has a long history in working on larval ecology, and he did some early seminal work on larval connectivity in blue crabs. And he has gone on to look at almost every aspect of larval ecology and linking them with population dynamics of marine populations, both invertebrates and fish. So if you are working with larval anything, or you should definitely have read the numerous papers cited by that are cited and worked by Steve Morgan. And what I've found interesting about his work is really connecting the larval ecology and the behavioral ecology with oceanography. And that he's going to talk about work today that includes that. And some of you in this room, or maybe people that have graduated, actually helped put some of the materials out for. It did not look like this, though. The day that some of the days they were sampling, so I'm going to turn it over to him, but it was not calm like that. But linking the ocean and marine systems.
Well, thanks, Diana, for the kind introduction. It's actually easy for me to remember when I was here as visiting scientist, and that was during the 97-98 El Nino when I was living on the sailboat in the harbor, walking down the dock, getting drenched every day. But yeah, thanks for the invitation. It's great to be back here. You know, it's been a while, and the last time I spoke here was actually then, and then so I've been by from time to time, and it's always nice to see the lab thriving. But as Diana mentioned, what I'm going to do today is talk about surfzone hydrodynamics as a key determinant of nearshore marine communities. And so I want to begin, as always, by acknowledging all the colleagues on this NSF grant. Alan Shanks helped with the biology. Jamie McMahon, of course, is right down the road as our surfzone hydrodynamicist. And Alt Rainiers did the modeling on the project. And then, of course, we have all the students who contributed mightily to this, and the assistants. And Z is here in the room with us today, so it's great to have her here. And she played a big role in all of the plankton work as well, and particularly the phytoplankton work, which I'll be presenting here today. Okay, so top-down versus bottom-up regulation of communities has been sort of discussed for a long time in ecology and marine intertidal ecology. And so initially, people put a big focus on top-down regulation of these communities through predation and competition. And then more recently, the focus, there's increased interest, shall we say, on trophic subsidies, food and larvae coming into these communities, and also regulating them through these bottom-up processes. So these subsidies might be rack and nutrients on the shore in the way of food, and then plankton, also for the filter feeders, and then larvae replenishing the adult populations. Okay, so these bottom-up forces are thought to create a latitudinal gradient and intertidal densities here along our coast. So as the story goes, densities are dense up in the Pacific Northwest. There's high larval supply, and because of that, there's little free space. And cascading down the road there, then you get strong competition and predation. Whereas down here in our neck of the woods, it's supposed to be the opposite, where there's limited larval supply, a lot of open space, and therefore weaker competition and predation. So what's less clear is whether this also applies to the subsidies of food and nutrients along this gradient. Okay, so why do we see this gradient in densities in the intertidal along the coast? So the prevailing view is that it is strong upwelling winds along our coast that are driving surface waters out to sea when combined with Coriolis force in this diagram by the seminal paper by Rough Garden. So winds come screaming down the coast, combined with Coriolis, surface waters go out to sea. Of course, many of the larvae are believed to develop in surface waters, so for a lot of people it makes intuitive sense that they're going to be blown offshore. And a convenient explanation is that the winds are stronger down here in northern central California than they are up farther north, and therefore there'd be a lot of larvae lost to the populations down here more so than up north. Okay, and then of course these waters are upwelled along the bottom, and you see the signature from this satellite image. I'm sure many of you have seen these things before, where the upwelling plumes of this cold upwelled water off of my neck of the woods up here around San Francisco Bay go far offshore, and so it's very conceivable that larvae would be transported off with it. And then if you notice, the gradient and upwelling shows up in the extent of these upwelling jets going offshore. So up here in Puget Sound, you see that that's not much of a problem, or south of Conception compared to Northern California. So the second tenet of this hypothesis is not only are the larvae lost at sea when and where upwelling is stronger, but then they can't get back to shore except during these upwelling relaxation events. That is, when these upwelling winds weaken. And so in that case, then the surface waters move back to shore and replenish these populations. So up in the Bodega region, where the winds are persistent and strong, then we have infrequent upwelling relaxations and therefore limited recruitment in that area, as well as down here compared to the Pacific Northwest. Okay, so that's the prevailing view. So I found pretty much the opposite in my work here, and so this is a different talk, but I'm just going to summarize it here. All the papers are sort of listed here, a lot of them anyway, that you can look up if you like. But there are three basic points that I have to make about this hypothesis. First of all, we've been out surveying plankton, entire larval communities, like all of the crustaceans that are in the water, like 46 species. There are all the other invertebrates that have been sampled as well. And what we've found, in contrast to this story, that the vast majority of these larvae are doing just the opposite. They're retained close to shore in a coastal boundary layer of weak upwelling winds and currents. And so they actually complete their entire development there. That's more than half of the species we've studied. Then there are other species, just as happens everywhere else in the world, that go set distances out across the shelf and come back. So you have other species that aren't retained close to shore, but they might migrate out to the mid-shelf and come back, or other species to the outer shelf and then come back, or still other species to the open ocean and then come back. So this is sort of a migration, onshore-offshore migration, that is very common in marine invertebrate larvae. Okay, so the other thing is that these migrations we see over and over in time and space. It doesn't matter whether we're in this windy place off of Bodega, whether we're sampling at an upwelling center, an upwelling jet, or in between them, or whether we're up and off of Oregon, where the winds are less. We see that these patterns are repeatable and reliable over and over every time we go out in time and space. So, and then the second tenet of this hypothesis is that they only recruit back during these relaxation events, which are limited in this area. Okay, so that's because the larvae are believed to be developing in the surface waters, whereas many species, actually recruit, not in surface waters, but in bottom waters, which are upwelled, so they can use this as sort of a conveyor belt, taking them offshore and then back onshore. The other thing is that all species don't recruit by just one single mechanism, by passively waiting for the winds to relax. You know, behavior is a key thing. There's directed onshore swimming, there's depth regulation on when they're coming back, there's internal waves, all kinds of processes operating in the plankton. So that's all I want to say about that. But this is just to sort of set up the story, you know, the motivation for the rest of this. So if this hypothesis isn't correct, then what is responsible for this latitudinal gradient along our coast? And that's what I want to address today. And so one idea is that it's the surfzone hydrodynamics that's really responsible for regulating recruitment along the coast, but also subsidies of food and nutrients along the coast. And so that's what we wanted to take a look at. And this then is reflected in shore morphology. And so, like most things in biology and physics, you've got these two extremes at the end of so-called dissipative beaches and reflective beaches. And I'm going to be using those words quite a bit today. So the dissipative beaches, quite simply, are broad, flat beaches with large waves that break on an offshore bar, and then the waves progressively roll inshore. Broad, wide surf zones, high water exchange, and potentially high larval supply. At the other end of the continuum, then you have reflective beaches, which are, there it is. Narrow, steep beaches, small waves that break directly on the beach here. Narrow surf zone, low water exchange, and low supply of larvae and food. Okay, so to test this hypothesis, what Alan Shanks and the rest of the gang did is we did an initial pilot study where we went out and did a classic road trip along the coast, and we stopped and looked at densities of barnacle recruits, limpet recruits along the shore at 10 sites from Oregon to Northern California. And here you see the data with barnacle recruitment on a log scale here. And then here are the suite of dissipative beaches, 5 of them, and 5 reflective beaches. And what you can see is that there are orders of magnitude more larvae on these dissipative beaches, that's the broad, flat beaches with the wide surf zones, than reflective beaches. And the same thing is true for limpets. So this is consistent then with this idea that surf zone hydrodynamics might be setting latitudinal gradient and densities along our coast. Okay. All right, so to examine this idea, these ideas more closely, what we did is intensive process studies down here. And 2 sites, one here at Sand City, I guess this is Sand City, just north of Monterey, and then Carmel River State Beach, which I'm sure you all know well, south of here. And then here's an overview of the dissipative beach at Sand City. Again, this is a broad surf zone. It has these alternating rips and shoals due to rip currents. And then it has this broad surf zone flat beach. And then Carmel River State Beach is the opposite. It has a reflective beach here. This dashed line shows you where the surf zone is, and see it's very steep here. So what we did as a group is we went out there for 30 days in June and July for 2 consecutive years. First we sampled the dissipative beach, then we sampled the reflective beach. And at each one of these sites we did hydrodynamics and modeling and plankton and settlement data. Okay, so just so you get a feeling for what I mean by dissipative and reflective beaches, and I'll be talking about these sites for a while, so just so you can get it ingrained what we're talking about. Here's Carmel River State Beach, the reflective site. On a rough day, that's a 10-meter wide surf zone. On a calm day, it's only 3 meters. Waves breaking directly on the beach, on this steep beach. So we sampled 3 sites. One is the beach itself, and then the rocks to the north and south of it. We took phytoplankton and settlement data. Okay, so how did we measure cross-shore exchange? Well, we had an array of ADCP and ADVs, which measure currents and waves. And then also we had dye sensors deployed along the shore here in the surf zone. And then here's a profile of the beach showing you where these sensors are located. This is mean sea level. Okay, this is what it looks like when you're sampling in the surf zone, Carmel River State Beach, the reflective one. So when you're doing these dye studies to look at cross-shore exchange and along-shore exchange, have a bunch of swimmers out there. They have GPS beanies on their head, and they're carrying around dye sensors. We do these fancy little drifter studies from kayaks just outside this mighty surf zone here. And then to sample plankton, we had to jet in these poles all the way across into the surf zone, and then had this plankton pump, and we just sucked plankton up from the surf zone. And then we sampled plankton offshore from kayaks using nets. And Z can tell you all about it. All right, so here's what the other site looked like. This is Sand City. So it has a 125-meter wide surf zone, more or less. So here you see it's much broader than the reflective beach. So here it is on a calm day. Again, the breakers are offshore, it rolls in. Here it is on a rough day, people sampling plankton out there. Give you a feeling for what that's like to be out there sampling every day for 30 days. And then also there's a system of rip currents and shoals in the system. So here are the shoals with the waves breaking on the bars, and then here are the rip currents going out, and then a shoal. This is important for surf zone hydrodynamics, and we'll be talking about that. This is how we measured cross-shore exchange at this site. Here's sort of an imaginary box that's set up, release dye in the center of it. We have two sorts of current and wave meters here. We have two boats out here with sensors. Also a jet ski armed with sensors that Jamie McMahon and the crew was riding around, mapping the dye. And then 7 swimmers out here with their GPS beanies and dye sensors. All right, and then 25 GPS drifters, and this is what it looked like in action with the dye release going across this broad surf zone. And then the plankton was done from a motorboat offshore, and then here's the GPS drifters. Okay, enough setup. So let's talk about, let's see some data now. And so we're going to start with the biology first to show you what we found, and then I'm going to use the physics to explain it. We're going to start with phytoplankton first. Again, this is work that was done by Alan Shanks with a lot of help from Z. All right, so here in this busy slide, you see that here are 3 sites of this reflective beach, Carmel River State Beach. And then here are the 4 most abundant taxa that we looked at. On the y-axis are log plots of the surf zone of phytoplankton. Here's the offshore. And that's offshore in quotes, because I'm using plankton collected 10 meters outside the surf zone. Okay. All right, so this dotted line is to indicate a one-to-one relationship between phytoplankton in the surf zone and outside the surf zone. So if it falls along that line, then that means there's free exchange going on back and forth. But what we see here for most of these taxa is that it actually falls well below the line, indicating that the higher densities are offshore than onshore. Again, remember these are logs, so these clusters of dots are sort of collapsed. All right, now the one difference we found was for Navicula, this species down here that showed the opposite pattern. This species lives in shallows in the surf zone, and this is the species then that's most abundant in the surf zone rather than offshore. So what you're seeing is sort of offshore species staying offshore, the onshore species staying in the shore, and if there was free exchange, then we would see it falling along this line. And in fact, we see very little correlation for most of these plots. So this is indicating that the surf zone is kind of a little bit semi-closed, right? There isn't a lot of free exchange in this particular shore. Now, if you go to the Dissipative beach at Sand City, we see the opposite pattern. Now, for the 4 most abundant species, as well as all of them combined, you see that they're now outside. So the highest concentrations are now inside the surf zone. So by outside, I really meant above the line. Okay, also you see that there's strong correlations from the inner shelf to the surf zone, indicating that these phytoplankton cells are being freely exchanged. Also, we sampled the rip currents and shoals. So here I'm showing you the data as just a daily record for 2 species. Well, I've been talking about the samples in the surf zone and these rip currents, which are really dense. We sampled offshore, 10 meters offshore. And now I just want to show you that the shoals are even less in the surf zone, so that you have structuring within the surf zone itself. Okay, so that's phytoplankton. Now I want to show you what we've found for zooplankton, which is easy because it's pretty much the same thing. Okay, so if you didn't catch it the first time, here we go again. But now the plots are different because we have more taxa we're looking at. So I just did these little histograms inside versus outside of the surf zone. So for this reflective beach, again, like phytoplankton, more zooplankton outside the surf zone than inside the surf zone. That's true for the holoplankters. Here are all the crustacean holoplankters like copepods. True for the jellies. It's true for all the diverse larvae of invertebrate species. It's true for a couple of the demersal plankters. But there were a couple of interesting exceptions. So here, taking a look at barnacle larvae. So initially, barnacle larvae showed the same patterns as all these guys. That is, they're more abundant outside. But then as they aged, you see there is now no longer a significant difference. And then when they now become post-larvae and they're ready to settle, you see it flips. So we have an ontogenetic shift horizontally from the inner shelf to the surf zone. But we also know that many barnacles have an ontogenetic shift with their position in the water column, where young larvae are typically high for many species, older larvae come deep. So this indicated that perhaps maybe these cyprids are getting back on shore by getting deeper in the water column. Okay, there is more circumstantial evidence for this, just taking a look at biological patterns. So harpacticoids, the benthic copepods, bottom dwellers, as well as parasitic isopods. Those are parasitic isopods coming in looking for adult hosts that are living on the bottom, and detritus, which sinks. All of these things that are near the bottom are the ones that are coming in, whereas the ones that are more up in the water column might be staying out. So that was kind of what these patterns told us initially. Now you go to the other beach, the Dissipative beach, and like phytoplankton, we see just the opposite pattern. Now, all of these taxa are more abundant inside than outside the surf zone. And also, as for phytoplankton, we see that all these taxa are concentrated in rip currents rather than on shoals, except for these weren't significantly different for the demersal taxa. Okay, so what we see is across this entire plankton community, phytoplankton, zooplankton, doesn't matter the species very much. You're seeing strong differences inside and outside the surf zone at these 2 different beaches. Physics has to be explaining these differences in large degree. Okay, so what might that be? Well, here's one thing that's similar at beaches worldwide, is that there's undertow, that is, surface waters are going out to sea, whereas then there's return flow of bottom waters coming in, known as benthic streaming. So this is a way then that most organisms staying high in the water column would be kept out of the surf zone unless they got very close to the bottom, and then that might bring them back on shore. I'm just showing you data for the reflective beach, but it's the same for the dissipative. Okay, now here's one difference between the beaches, and that these reflective beaches have much less cross-shore exchange, and this was documented by these physical sensors and dye studies that were done by Jamie McMahon and Jenna Brown, his student. So here at these reflective beaches, it's really pretty much like a large swash zone with standing rather than progressive waves. By that I mean that these waves are coming up and they're breaking directly on the shore, and then the water rolls out, and then the next wave comes. And so it's just oscillating like this. So you can imagine that there might not be a lot of opportunity for exchange with the surf zone from further offshore. Okay, so to take a look at this, of course, they did the dye releases, and here's the dye being released. Here you see it rapidly spreading across this narrow surf zone and then down the beach, but it's not spreading out to sea much over time. So what they found is that the exchange is about 50% of that of dissipative beaches. So that might explain why the plankton are staying offshore rather than onshore at this beach. In contrast, we see the opposite thing for plankton going on at the dissipative beach because the physics are different. Here, remember, we had rip currents, and here's a diagram showing the winds and waves are bringing plankton in over the shoals, and then they go back out to sea in these rip currents, and it recirculates like this, setting up these recirculation cells or eddies, which might be a way of exchanging water and plankton from outside the surf zone to inside the surf zone, and then just recirculating it, concentrating these cells, which would explain why we're getting higher concentrations. So here's data from Jamie McMahon showing the vorticity, fancy word for spinning. The orange is the faster spinning, and this is showing the spinning in these recirculation cells. And then these black arrows are GPS drifters being caught up or entrained in these eddies and spinning around. Okay, so that's the biology and the physics. And then we needed to have a better idea of what the mechanisms were for exchange or lack thereof of these plankton into the surf zone. And so for that, we turned to individually based numerical modeling, and this was done by Ott Reniers and his student Atsushi Fujimori from University of Miami. And so I'm just, here are the take-home messages from this work. That more zooplankton was delivered into the surf zone at the Dissipative beach. And there are two ways for zooplankton to cross surf zones at both types of beaches. Okay, one is deep via this benthic streaming that I was talking about earlier that cyprids might use. And that only occurs if there isn't a lot of wind, because with big wind comes big waves, which is a lot of turbulent mixing, and it disrupts this benthic streaming process. Okay, the other way, of course, is to stay near the surface and have onshore wind forcing and Stokes drift, fancy word for wave transport, bringing them onshore. For each of those mechanisms to work, they found that it had to be coupled with a behavioral response, and that is sinking in response to turbulence. And there have been a number of studies done in the laboratory with diverse invertebrate larvae showing that when there's turbulence, like you might get as you're crossing the surf zone and the breakers, that organisms sink to the bottom to escape the turbulence. Okay, so in these models, they had three important variables: depth preferences of the organisms, sinking in response to turbulence, I was just talking about, an onshore wind with Stokes drift. So here's some model outputs for the two beaches, the dissipative versus the reflective. Here are the two mechanisms I was talking about: no wind, stay deep, sink in response to turbulence, and the benthic streaming carries you onshore, versus wind and waves bringing you in at the surface. So here are the model results. So all these simulated plankters are released outside the surf zone. And then we just let the model run and see what happens related to the physics. And you can see from these orange-red colors that at the dissipative beach, there's high concentrations of these plankton by both mechanisms on the dissipative shore. And in contrast, at the reflective shore, you don't see much being concentrated in the surf zone, indicated inside this dotted line, hardly anything. All the concentrations are here in little eddies that are setting up offshore. Okay, so take-home message. These model results with these two mechanisms show, reflect what's going on well in the field. All right, just to show you how sinking is really important in this model, taking a cross-section for these two kinds of mechanisms of onshore transport. Just going to show you the results for the dissipative beach, but it's the same for reflective. So here, they're coming onshore deep by benthic streaming. Then they get to the surf zone and they can't cross it without sinking. But if they sink in response to turbulence, that keeps them there in that benthic boundary layer, and they're able to cross. Okay, and then same thing with wind. They're coming into the surface. Without sinking, they don't get across. With sinking, they sink, and boom, and benthic streaming carries them onshore. Okay, also, the last thing is that the model shows quite well at the dissipative beach that both of these mechanisms concentrate plankton and these rip currents, which we found well in the field. Whereas at the and then here's some example particle tracks of this happening, so you can see them coming in along the bottom in these deeper rip channels and recirculating in here. And the same thing happens if they're coming into the surface, they sink down, and then they're sort of entrained into these rip currents. Also, there's some sort of entrained in little eddies at the surface due to, wherever these rip currents are exiting the surf zone. Okay, so that's how these mechanisms work. Okay, so you've got the two kinds of extremes of beaches, at least in concept we're talking about, and processes for what might explain these broad patterns. Now I want to return to the original question I set up and take a look at this latitudinal variation in larval recruitment. Remember I said the prevailing hypothesis is upwelling winds is not really explaining the patterns that we see on the shore, at least by the processes proposed. Now I wanted to look and see if actually surf zone hydrodynamics might explain these patterns along the coast. So what we did, classic road trip, 42 sites along the entire extent of the west coast of the US from San Diego to Puget Sound area. 42 sites, 17 of them were boulders, that's rocks and sand versus more bench type things, in other words, extended rocky habitat. And our sampling differs from these three studies that looked at this question before in a couple of ways. One is we're covering the entire coast. They covered pretty much Point Conception to northern Oregon. And also we're doing 2 to 3 times as many sites as the previous studies. Okay. First, I want to show you the potential players in terms of physics that might explain latitudinal gradient and densities along the coast. So, of course, if you're thinking upwelling, then you will want to concentrate on wind stress, and waves might be associated with strong winds. And yes, there is latitudinal variation, but it's not exactly what you would expect to explain higher concentrations in the Pacific Northwest than down in Southern California, because it's dome-shaped. Why? Because it's been well documented that it howls where I live in Bodega Bay in Northern California, but it drops off. So you don't have sort of this steady increase that you would expect to see. I'll talk more about that later. Same thing with waves. On the other hand, here are 3 common surf zone hydrodynamics metrics. Surf zone width, remember I've been talking about from these 2 beaches. Intertidal slope, how steep they are, and also intertidal width, that is how wide these beaches are. So what we see from the data is that you have more dissipative beaches, that is broad surf zones, flat beaches, wide beaches up north. Okay, so there's a potential for seeing surf zone hydrodynamics affecting densities and the intertidal. All right, let's take a look at the data. So I've broken up the data into sort of juveniles and adults versus recruits. Again, barnacle density on a log scale. Here are the different metrics, physical metrics. So start with latitude. And you can see that there is a relationship with density of barnacles along the coast. But notice that it's actually, keep going for the big button, it's actually quite weak. It's not strong at all. Okay, so it's there. I don't know how much to make out of it. But when you take a look at wind stress, i.e. upwelling, there's no relationship whatsoever with our sites, nor is there for wave height. And this is true, for the boulders and the benches separately or combined. Okay, so here the surf zone hydrodynamics. I'm just using surf zone width as one metric for the rest of the slides. Be the same for all of them. Again, broken up log barnacle densities for juveniles and adults, recruits, and percent recruits. And what you can see is whether we take a look at boulders, benches, or all data combined, we see very significant correlations in terms of densities all along the coast and surf zone metrics. Okay, so in other words, surf zone hydrodynamics appear to be regulating populations along our coast. Okay, that was in space, so we wanted to take a look at this more closely in time. So we selected some of these sites and we monitored them weekly, 9 of them from 2 to 3.5 months. For 7 others, we looked at them daily for 30 days. Again, log recruitment along this line. Here is our surf zone width metric. Here are the results for the weekly sites, and you can see that there's a strong relationship over time, and it's even stronger still for the daily measurements. So again, surf zone hydrodynamics appear to play a big role in regulating intertidal populations in time and space. And the last thing I talked about was rip currents, right? So now I want to go back and say, well, are rip currents a big deal along the coast? So rip currents occur only where the surf zones are greater than 60 meters wide. So here's rip currents, here's surf zone width, here's number of rip currents. And you see nothing but zeros less than 60 meters. So rip currents only occur on these broader beaches, i.e. dissipative beaches. Okay, and of course, what do you know about dissipative beaches? That's where you get most of the larvae recruiting. Okay, so again, association with rip currents, dissipative beaches. Okay, now there's another way of looking at this. This is the so-called surf zone retention parameter, which is just the surf zone width versus the spacing of the rip currents within the surf zone, and that gives you an indication of the intensity of these upwelling of these recirculation cells, concentrating plankton and other matter, and you can see that there's very strong relationships, again indicating the importance of rip currents. Okay, so that should bring a question up in your mind. How come we found something different than all those other guys? Okay, so we worked with Sean Connolly, who was kind enough to provide information to us, so we go back and revisit the way they actually did things to sort of sort this out a bit. Okay, so here you're taking a look at latitude, percent cover of barnacles that Sean measured. Here he started in San Simeon going through northern Oregon, and here are his data and these triangles. And notice that it's low in California. And then when you go north of Cape Blanco, indicated by this dash line, there's a big jump up in densities. And I should mention that he's getting these data by using quadrats on the rocky shore, and he's measuring algae and barnacles and mussels. And so these are natural data of entire sorts of populations. I'm just showing the data for percent cover of barnacles. Okay, so plotted here is upwelling strength, this alongshore wind stress. And again, we see this dome-shaped curve. Okay, so you wouldn't expect then for these to match well, and in fact, there is no correlation. But they did find a correlation. Why is that? And here comes one of the reasons for why their data may differ from ours, their conclusions may differ, and it is they took wind stress from the mooring that's out near the Channel Islands where it's howling and it's really windy, whereas all their sites are on the mainland in the cozy Santa Barbara Channel where the winds really drop off. Okay, so, and point of fact, you know, their data would be more up here in the south, in which case you would get a strong correlation. The other possible explanation is we did the entire coast with 2 to 3 times as many sites. So anyway, when you take his data and now you map it onto Surfzone width, we see that there's a very strong correlation again. Again, indicating that Surfzone hydrodynamics may play a big role setting densities in old data sets as well as new ones. Okay, also the second study they did was they went pretty much to the same sites, and this time they used settlement plates, standard Pisco style, PVC, safety walk tape on top of them, set them out, come back, measure recruitment. Very nice study. And here you see his data matching up well with natural populations, which I showed in the previous slide. But I've also plotted on here solar radiation. Of course, it's sunnier down here than it is north of Cape Blanco, duh. And so there's actually a strong correlation with solar radiation and settlement on the shores. So we've got a couple of things going on, Surfzone hydrodynamics and also potential post-settlement mortality from heat stress. Alan Shanks has already shown in a previous study that these settlement plates get up to be 40 degrees C in bright sunlight, so you have more of that California than north of Cape Blanco, which might explain these results. The other thing I sort of skipped over before that's important in a previous slide on Surfzone hydrodynamics is that we have a very strong relationship in that percent recruits graph of the number of recruits correlates very nicely with the adult populations, which indicates that it's larval supply that's establishing these populations rather than post-settlement mortality. Sorry for skipping over that panel. Okay, so this brings me to the conclusions. So you can read along with me here. Different Surfzone hydrodynamics at reflective and dissipative beaches affect cross-shore transport of zooplankton and phytoplankton, entire plankton communities. So there are pervasive, large differences cutting across wildly different taxa, behavior and no behavior. And so at reflective beaches, concentrations are really sparse in the surf zone. There's less onshore transport. And at the dissipative beach, it's just the opposite, where concentrations of plankton are dense. We talked about the physical mechanisms of the reflective shorelines. We have low water exchange, which limits onshore transport. At the dissipative shores, we have these recirculation cells from rip currents concentrating plankton. And the modeling showed that plankton may cross the surf zone in 2 ways. Remember, deep by benthic streaming, sinking in response to turbulence that only happens under calm conditions. And then the other way they get on shore is when it's windy, coming in by winds and waves at the surface, again coupled with this sinking. All right, we did our little road trip, and we found that coast-wide densities of barnacles and recruits are related to surf zone hydrodynamics. Densities were not related to upwelling, and this proposed latitudinal grading in densities seems to be quite weak in our data set. All right, so here's my take-home message, boiling all this complex stuff down into one easy sort of thought. So surf zone hydrodynamics regulate larval and food supply to these nearshore populations and likely are a key determinant of community structure and dynamics. And it's these bottom-up forces that is not just larvae, but food coming in that's regulating these intertidal nearshore populations. But it's not the way people have been talking about it, upwelling, upwelling, upwelling on the brain. It may, in fact, be surf zone hydrodynamics. And that's my take-home message. Thanks. Yeah, Mike.
So when you go down to a rocky shore, especially if it's expansive, and I think some of your sites are like that, how do you characterize the surf zone?
Yeah, so let's see, this is a key question. And so all the classic work in surfzone hydrodynamics has all been done on beaches. For obvious reasons. It's really dangerous and treacherous and difficult to do on rocky shores. And that's why we did our process studies to take a look at this on beaches with rocks and jetties, so we can take a look at sediment. So really the next step in this journey is to try and do the same sorts of studies, process studies on rocky shores. So we don't have a good idea. I don't have a good answer because there's no information on this as far as I'm aware on this, except to say that in general, I think maybe these rocky shores are going to be more like reflective shores and they're more likely to be steep and hard and all that. So the only information we have showing this works for all different kinds of rocky habitat is we separated out in the latitudinal survey, separated out water fields from rocky habitat and results from this. So to characterize it, I would say that there's going to be more reflective in this. Enough?
So when you went back and revisited commonly sites, so how did you then characterize those as a core? I mean,
Right, so what we used as a metric is rocky sites, those sites that were 50 meters or so expanse at least, not much, if it's going to be rocky, it's got to be 50 meters and not much sand on either side. So we had to find a way to classify these things. I guess this is your question. So thanks for asking again. And to sort of categorize these as into rocky versus more sandy.
A little bit touching up on what my guest, I'm thinking about the scale of each process, or you talked about the scale of each process, this beach is reflective or dissipative. But if you went out to a continuous shoreline or something that had a mix of these things, do you have an idea of the spatial scale at which these processes play out?
Well, let's see. So I guess that it just depends. So it just depends on the extent of it. I mean, it's hard to talk about conception. We had a little map which we had in mind to say, well, can we surf zone hydrodynamics really set up in this really combined scale? But clearly, I think as we moved along the shoreline, we've been closer to space sites. I know we're closest to it, but we're talking about meters, kilometers, tens of meters. I think it would be difficult for these processes to really show up tens of meters, but you'd probably be able to see it from cove to cove or beach to beach. So it's a whole surf zone hydrodynamic system.
Steve,
I'm working my way back.
I'm having a little trouble.
I'm doing murder as well.
I'm having a little trouble teasing apart some processes related to source and sink, and related to the stage of plankton, whether the early life stages of plankton or later, more relative to settlement. So I guess my question is, I understand the process and how it affects whole plankton. It gets swept in, it can be food for things. But meroplankton, there's got to be an influence on where the source organisms are that are producing plankton, number one. And at the other end of the whole process, what their objective is, which is to settle somewhere. So that little demersal corner on one slide struck me because those look like larvae that are looking for a place to go. They're not looking like they're from a place where they originated. And necessarily. I'm having trouble with that. So do you distinguish life stages of these planktons, or?
Yeah, we, as if in the past, painstakingly identified every stage. I'm sure you have it up there.
Okay.
The results are the same across pages except for the ones that pointed out like the lines. So that's the prime example that shows how they stay outside if they behave like phytoplankton, whole plankton, staying high in the water column. It's only when they move deep in the water column that they can penetrate. So I was going to say, how do I get on shore? That would be a prime way to do it, is to move deeper in the water column. So I think we see that. And so, of course, the only stuff that's going to come on shore that happens to be in the neighborhood, right? So, I keep talking about beaches and all this stuff. There's rocks on these beaches. So it could have come from there. Chances are they probably didn't. It might come from just around the corner, just up the shoreline. They're there now. And they can probably smell barnacles on the rock and stuff that come in.
Yeah, so I actually had two simple or straightforward but linked questions, but following off of Greg's. So part of the big idea with rough garden colony is this idea of a larval pool. Are you suggesting for those species that have a long duration in the plankton, they never actually make it there and they're staying within the surf zone? Or is the surf zone a way to get to the larval pool and back independent of upwelling? I don't know where the timing is. Clearly for something that's in the water for a week, it's different than something that's in the water for 6 weeks.
Yeah, so, really, but my sense is that all these organisms are going to move out of the surf zone and then come back. Many species are going to develop near shore, and then they're going to be held up from getting back onshore, depending on whether they happen to be near a dissipative shore or a reflective shore. And so almost all larvae are designed to disperse initially away from adults, so they tend to get up high in the water column where the currents and waves and winds are going to carry them some distance away, not too far. And then you see behaviors that set up and enhance feeding, attention, and then ultimately, for the host larvae, their job is to get back home.
And then another part of the Connelly work is you can think of it as a gradient, you can also think of it as a step from high latitudes to low latitudes, and just a high recruitment area and a low recruitment area. And in the high recruitment area, you can have a correlation between your dissipative beaches and upwelling, right? In the sense that their relationship falls apart at low latitudes. It really does very badly down in Southern California. Is it possible that the processes they're talking about really work themselves through in Central California such that you have more of an Ali effect? That once you get to Southern California, there's just not enough barnacles or mussels around to really provide the source to come back to the beaches, and that all the upwelling dynamics really happen more to the north and are still relevant there, but are just not relevant in the south anymore because of the changes in the adult densities that are supplying to the larval pool in the first place.
Yeah, well, first of all, they didn't really sample Southern California. They sampled Central California to Oregon. And so, is your question?
I guess how big is as you move south, you're losing adults on the beach, or basically on the rocks. Therefore, regardless of the process, you're going to have less recruitment coming back to it. Anyway, so maybe upwelling is really happening at their more northern sites, they're dynamic, and as you get south, it becomes an irrelevant process.
Let's see, I guess I'm not entirely following you. I would just say that processes are going to be the same regardless of where you are along the shoreline. So you have your upwelling gradient, you have your surf zone, hydrodynamics, sort of characteristics are working all along the beach. And remember that there isn't much of a gradient according to our more extensive views. There is one, but it's really more about what the characteristics are right there and next to the shore that's between. So, I think there's very little evidence that upwelling is working for this, and I guess I'm not really answering your question because, I think it would be true whether there's limited larvae down there or not. This is just fundamental mechanisms that are not shown. And you can can explain it to you afterwards. I'm going to do a better job.
Steve, you mentioned that sinking was like a behavior almost. Is that another way of saying they actively did not swim?
So there's two ways to get to the bottom. One is to stop swimming, the other one is to actively swim. So there are larvae, for instance, oyster larvae. If you give them a chemical cue that they hit when they're swimming over an oyster bed, they dive on, so they can hit that reef for a bit, that pouch before they get. So I think that what you call it sinking, this is a model. You just gave it a rate moving down. It wouldn't terribly sinking up. So anywhere you get the bottom is going to be this mechanism.
But the mechanism is would be to turn off swimming and just rely on your natural specific gravity to get you down.
Or to swim down.
But, and you were basing that on an idea or a measurement?
So basing that on the fact that larvae and the lab, multiple species do that. And so it happens, but we have no documentation in the field that that's actually going on.
And the link to turbulence was that, now in the Surfzone, I have to imagine that everything is isothermal throughout your whole study area, and there's turbulence everywhere, and what you mean is that massive turbulence causes something to stop the swimming of the organism so that their specific gravity takes them to the bottom.
Yeah, I think when they hit the breakers, the Surfzone, that's the turbulence.
That's the turbulence.
Yes, Rick.
So, that's an interesting description of what's going on in the Surfzone and the intertidal. What do you see in the shallow subtidal? That might be rocky subtidal. Do you think the processes are going to be similar or completely different? Just outside the Surfzone and 30, 40 meters deep or something.
I think it's going to be very different. So I think you can all of the work that has been done in the past would apply out there. There's been very little work done on larvae in the Surfzone. And I think this is kind of the stuff we're talking about here. I don't think what I'm talking about the frogs on the Surfzone. All right, you students. Somebody, please. Yeah. All right, you lost your turn.
So, I recall a few years work on urchins that we did people based recruitments around headlands and stuff like that. So I was curious, it just occurred to me, based on what you said, well, headlands are often maybe at the ends of dissipative beaches. Is that? I wonder if there's a correlation between headlands and dissipative beaches for occurrence rather than something to do with alcohol.
No, if there is or not. It's an interesting idea, and viewers would find the importance of headlands in recruitment, so that's an interesting idea for. Yes?
I have a question about how El Niños would affect this, because you would expect that if upwelling is kind of the key to larval recruitment, then it would kind of break down during an El Nino when you get the warmer water. So do you think your mechanism would explain recruitment better regardless of oceanographic conditions?
Well, let's see. So, some pure speculation, of course, but I'd say during El Nino, you're going to have waters that are coming up particularly strong forward flow, and there might be a little bit more onshore transport. So it's possible that you could get a higher recruitment onshore, maybe more water exchange. I want to say that was Surfzone. I don't know. That's where we need Jamie McMahon and okay, stump me.
I want to thank Stephen